All right, so I want to talk to you about meiosis and crossing over and how they occur and what it looks like. And so what we're going to use is this diploid cell here. A diploid cell contains a full set of chromosomes. A full set in our case will be a total number of 4 chromosomes, 1, 2, 3, 4. So before a cell goes into meiosis, which carries the same stages of mitosis of prophase, metaphase, anaphase, and telophase, before it goes into that, it needs to replicate its DNA. And it does that during interphase, during S phase of interphase. So we're going to show the replication of the DNA during S phase of interphase here. So here we go, replicating that DNA. And that one, and that one. So we just went from 4 chromosomes to 8 chromosomes. Now, the next thing that happens is we're going to go into meiosis. And during prophase of Meiosis I, which we'll call Prophase I, the homolog chromosomes come together. Now, we're going to signify the pink as mom and the yellow as dad's chromosomes. And so we're going to go ahead and bring them together here. Now, this forms a tetrad. A tetrad, tetra means 4. So we have a tetrad is 4 chromosomes together here. And they get so close that they actually start to intertwine and form a synapse. A synapse is where we see the chromosomes, those sections there, touching. And we do the same thing over here as well. Now, this is crossing over, and what can occur here is an exchange of genetic information. So I'm going to pop up a few beads and exchange them to show you what I mean here. Take these two here, and these two here, and exchange them. And I'll go ahead and take this one here and this one here and exchange them. Okay, so this occurs through in prophase I. Now we go into metaphase I, and the cells will line up in the middle here. Okay, whoa. And then we'll get into anaphase, where they will be pulled apart into opposite ends of the cell here. And then we go into telophase and cytokinesis, and we will create 2 new daughter cells. That was Meiosis I that just occurred. Now we go into Meiosis II. In Meiosis II, there is no DNA replication and there is no crossing over. Instead, we go into prophase, metaphase, anaphase, and telophase again. So let's show that. Line them up in the middle for the metaphase. Go into anaphase and pull them to the opposite ends of the chromosomes, to the opposite ends of the cell. Same thing over here. And then we go into telophase and cytokinesis. And now we've just created our 4 haploid cells. They're considered to be haploid cells because they contain half the amount of chromosomes as that original cell. That original cell had 4. And now we have 2 in each. So they have half the amount of chromosomes. What is this useful for? Well, these are our sex cells. These are our gametes. And so if you're a male, you'd be creating 4 sperm. If you're female, this would create 1 egg and 3 polar bodies. And I get into that in the previous presentation over meiosis that is on the classroom. So let's also take a look at the differences between all of these and what crossing over has given us. We have 4 genetically different cells. Without crossing over, we would have just clones. But now we have 4 genetically different. How do I know that? Well, take a look. the patterns of the beads are all different from one cell to another. None of them are the same. So that means if this sperm was to fertilize an egg, then it'd be much different, or just a little different, than if this sperm was to fertilize that same egg. This gives us genetic variability. This is why we all kind of look a little bit different from each other, why you don't look exactly the same as your brothers and sisters, is due to this crossing over that has occurred. Genetic variability is also very important for a population of a species for evolutionary purposes. If we were all clones, and let's say a disease came into the population, and we're all the same, and no one had some type of genetic variability of a built-in defense, then the entire population might fall. But if somebody or a couple people have a genetic variability that gave them this innate defense, this adaptation to that disease, then they will survive and they will reproduce. And we'll talk more about that in natural selection and evolution next semester. But I hope this helps you understand the process of meiosis, going from the first diploid cell, replicating the DNA, crossing it over, splitting the chromosomes into the two new daughter cells during meiosis I, and then again splitting them again in meiosis II to create these 4 genetically different haploid cells. If you have any other questions, let me know in class or let me know in the comments section of this video.